Hello everyone, my name is Anfam and I am going to present to you about the tight junction. About the background, tight junction is a type of cell-to-cell junction that exists in animal cell. Tight junction composed of many bands which are closely adjacent to each other and form barrier between the internal cell of the body and the extracellular environment. So think about if you are building a wall, how are you going to link every brick together to build a wall? In this case, cement or concrete would use to bond between brick and seal the inside with the outside wall. In animal cell, tight junction play a very similar role as the cement in the wall example. In plant cell, this phenomenon is known as the cell wall. So, in fact, the tight junction play an important role in the regulation of the passive epithelial movement of molecule. It can act as a border control that can select which ion or molecule can enter the cell and which must be kept outside of the cell. Several signaling molecule has been involved in the regulation of tight junction functions such as calcium protein kinase and G protein. For example, the change in intestinal permeability of the intestine are related to the alteration of tight junction as an expression of intestinal barrier damage. Let's talk about the structure and protein involved. In tight junction structure, cells are held tightly against each other by many individual groups of tight junction that protein called the claudine. They are almost they are the most apical component of the epithelial junction complex, and they are also connected to the actin cytoskeleton. Many proteins are special involved in the tight junction, including the cytoplasmic actin binding protein and the adhesive transmembrane membrane protein. In this figure, show an experiment of tight junction testing with a dye solution to see if the solution can diffuse through the tight junction barrier. As the result, the tight junction were doing their job, which prevent the solution from getting inside the cell, which they stop the solution right here. Where can we find the tight junction in our body? Tight junction are especially dominant in the intestinal epithelial cell. They are also abundant in the bile duct and the cavity of glands that connect with the digestive tract, such as the liver and the pancreas. Moreover, urinary bladder, where they function to keep the urine from leaking to the cell. In this figure, the blood vessel of the brain also bound by a tight junction, which play an excellent role in protecting the blood vessel and help balance neuron function, also known as the homeostasis. Disease associated with malfunction. Since tight junction was dominant in the epithelial cell, there are many diseases that relate to the epithelial cell, such as the junctional epidermolysis bullosa disease. A mutation in one of the subunits of laminin caused the outer layer of epithelial cell and underlying tissue fail to form properly. Therefore, patients with this disease would develop a blister in large portion in their body. Some primary research on tight junction that I do is the intestinal permeability regulation by tight junction, implication on inflammatory bowel disease. About the disease is the tight junction is a multi-protein complex that form a selective permeable shield between adjacent epithelial cell and dermis cake, the boundary between apical membrane domain. So the disruption of the intestinal tight junction barrier followed by the permeation of luminonoxus molecule induce a perturbation of the muscular immune system and inflammatory, which act as the trigger of the development of intestinal and sy- system, systemic disease, inflammatory bowel disease. Patient demonstrate increased the intestinal paracellular permeability, although it remains unclear whether the barrier dysfunction precedes disease or this result from active inflammation. Increase in intestinal tight junction description is observed in the IBD patient, such suggests that this regulation of tight junction barrier integrity may predispose or enhance the IBD procession. So my question about this topic is how the tight junction can be distinguished from other cell-to-cell junctions such as adhesive junction or cell wall. Here's my reference. Thank you for listening.